I've thought about this a lot, and I think it just really just comes down to two things, and it's the solving the machine brain bandwidth constraint and democratization of AI. I think if we have those two things, the future will be good.
Like, we didn't evolve to have a communications jack, or some. So there's got to be essentially vast numbers of tiny electrodes that are able to read write from your brain. We've got to build an interface.
The mission of Neuralink is to build affordable and useful brain-machine interfaces that merge humans with AI and solve blindness and paralysis in the process. This generates headlines and grabs eyeballs, but conversations online tend to be more about the aesthetics of being a cyborg than the technical development of the devices. Is it cool or is it creepy to have chips in your brain? These conversations also revolve around philosophical implications of the future fully realized end state of such a technology. Will it allow us to fully merge with AI and mitigate the alignment problem? What would it be like to share mental states with your friend rather than having to use language to speak to each other? The technical conversations are mostly limited to the demos of surgical robots in action or monkeys controlling computers with their brains. The demos make the Neuralink feel tangible. You can see Pager the monkey with a chip in its brain, playing a video game with his mind for a banana smoothie. You can see the surgical robot sewing wires into a fake brain substrate. This is, of course, insanely cool, but in order to understand the real technical action that makes these demos possible, your mind has to be comfortable thinking at the scale of micrometers and moving laterally between biology, material science, electronics, and software. This is an alien landscape for most of us. Most of us simply are not used to thinking across such a wide range of tiny systems like this. Our intuition breaks down without the background knowledge. I suspect that many people who find Neuralink interesting have gotten lost and given up in trying to understand how the link actually works. If you are one of these people, this series of posts is for you. I'm a neuroscience PhD student and a huge fan of Neuralink, so I thought to challenge myself to explain the technical stuff, including those that are outside of my comfort zone, in a way that is concrete and easy to understand to anyone who is curious and willing to make an occasional trip to Wikipedia or YouTube. The point of this first series of Neuralink posts is to break down the original Neuralink paper, published in 2009 by Elon Musk and Neuralink, titled An Integrated Brain-Machine Interface Platform with Thousands of Channels. The original paper describes the prototype Neuralink device, surgical robot, and software stack for interpreting the brain's electrical signals that existed at the time of publication. Since that paper was published, Neuralink has iterated on the original prototypes and shared major updates in several public presentations. So the information in the paper is certainly outdated, but it's still comprehensive of the whole Neuralink system, and so it works fine as an entry point to understand the technical problems Neuralink is trying to solve and how the team is going about trying to solve them. The paper covers a broad range of science and engineering, from material science and microchips to neurons and the immune system. The whole picture presented in the white paper also provides a vantage point from which to compare any future iterations, which gives your perspective some parallax to compare the whole endeavor and how fast it's progressing and maybe why they've made certain changes. In a future post, I'll talk about the device in a broader context, the company's long and short-term clinical goals, the economic need for manufacturing efficiency and quick iteration cycles, and updates to the hardware that have occurred since the white paper. But first, the main focus is to get through the 2019 white paper. In order to get through the whole paper, I'll read directly a section from that paper and then provide a kind of layman's translation of it, where you don't need to be a neuroscientist or a computer engineer in order to follow the plot and understand the problems that the Neuralink team is trying to solve. So you can skip forward past the parts where I read directly from the paper if you just want to have the translation of it effectively. Brain-machine interfaces have the potential to help people with a wide variety of clinical disorders. For example, researchers have demonstrated human neuroprosthetic control of computer cursors, robot limbs, and speech synthesizers by using no more than 256 electrodes. Although these successes suggest that high-fidelity information transfer between brains and machines is possible, development of brain-machine interface has been critically limited by the inability to record from large numbers of neurons. Non-invasive approaches can record the average of millions of neurons through the skull, but this signal is distorted and nonspecific. Invasive electrodes placed on the surface of the cortex can record useful signals, but they are limited in that they average the activity of thousands of neurons and cannot record signals deep in the brain. Most brain-machine interfaces have used invasive techniques because the most precise readout of neural representations requires recording single action potentials from neurons in distributed, functionally linked ensembles. Microelectrodes are the gold standard technology for recording action potentials, but there is no clinically translatable microelectrode technology for large-scale recordings. This would require a system with material properties that provide high biocompatibility, safety, and longevity. Moreover, this device would also need a practical surgical approach and a high-density, low-power electronics to ultimately facilitate fully implanted wireless operation. Most devices for long-term neuronal recordings are an array of electrodes made from rigid metals or semiconductors. Although rigid metal arrays facilitate penetrating the brain, the size, young modulus, and bending stiffness mismatches between stiff probes and brain tissue can drive immune responses that limit the function and longevity of these devices. Furthermore, the fixed geometry of these arrays constrains the populations of neurons that can be accessed, especially due to the presence of vasculature. So, the introduction describes the potential of a high bandwidth electrical interface between brains and computers and the current limitations of this technology. The factors that currently limit the utility of brain-machine interfaces falls into two categories: the fidelity of the information being exchanged between the brain and the machine, and the biocompatibility of the implants. Naturally enough, Neuralink aspires to solve these problems, building high-fidelity interfaces that remain safe and functional for a long time. For Neuralink's purposes, high fidelity means as spatially and temporally precise a readout of action potentials as possible. Neuralink cares about reading and eventually writing the action potentials of neurons because most modern neuroscience takes for granted the assumption that Francis Crick famously called the astonishing hypothesis, namely that the content of the mind and behavior are encoded in patterns of neuronal activity. This assumption underlies fields like cognitive neuroscience and behavioral neuroscience, in which one can experimentally manipulate the activity of populations of neurons and affect memory, behavior, emotion, etc. Action potentials are a telltale sign that a neuron is passing signal along to the next neuron it's wired up to. And so the decision by Neuralink to focus on action potentials makes perfect sense within this standard materialist paradigm of modern neuroscience and cognitive science. Neuralink aspires to detect the action potentials with high fidelity, but what does high fidelity mean in the case of a brain-machine interface? To understand, let's compare two fictional brain-machine interfaces, one that's low fidelity and one that's high fidelity. First, let's look at this old school electroencephalogram looking thing that Eleven is wearing in the scene in Stranger Things. Assuming these little brown bits on the array are electrodes, the electrodes on Eleven's EEG are positioned to detect changes in an electrical field that reaches through the skull and across a couple of square centimeters of the skull's surface. This field is huge compared to the size of the field of an individual cell. In fact, the signals detected by an electrode here in Eleven's device is likely created by millions of neurons. This device therefore detects changes in the average current across a large population of cells, with a sampling bias toward the cells that are closest to the electrode. Eleven's EEG is an example of a low-fidelity electrical interface. Such a device is incapable of detecting individual action potentials but it can detect waves of electrical potential changes that occur when whole brain regions are recruited. So this is probably more detail than we need to go into now, but you can detect, patterns of excitation and then depression in whole areas of cortex fire. Elon Musk compared surface EEGs like this to holding up a stethoscope to the outside of a football stadium to figure out what is going on in the game that's going on inside. These are useful tools for listening to the activity of whole brain regions, but any information that is encoded in the activity of the more granular subpopulations of that brain region is lost. Compare Eleven's EEG to a more futuristic science fiction device, the neural lace from Ian Banks' Culture series. A neural lace is a diffuse machine mesh that grows symbiotically within a human brain from prenatal development all the way through adulthood, where it is fully integrated into the entire brain and provides an instant readout and control of every single neuron there. This kind of brain-machine interface represents the limit case for high fidelity. It's perfectly aware of all cells at all times. This limit case might be an impossible goal, but comparing the EEG from Stranger Things and the Neuralink from Culture, Neuralink is definitely aspiring to build something closer to the Banksian lace. For a high-fidelity readout of neuronal activity in the real world without this kind of symbiotic machine mesh that grows along with the developing fetus and child, electrodes have to be implanted in the tissue so that the electrical field of each electrode interacts with a localized small population of neurons, as few as one cell per electrode. Because of the tiny domain of each electrode, detecting the high-fidelity signal from a section of brain tissue requires a high density of these electrodes. The optimal electrode density is up for debate and will probably end up being case-specific for the intended use of the device, and indeed, the Neuralink white paper includes multiple designs for electrode clusters with variable spatial density and orientation. The other major limitation of brain-machine interfaces is biocompatibility, which is a fancy way of saying the safety and functional longevity of the implant. The problem of biocompatibility cannot be understated. It's huge. If this is solved, then brain-machine interfaces become much, much safer. However, the goal of biocompatibility is the goal of reaching into the future and pulling out an alien-level, S-tier, almost magical technology. It's physically possible, but it requires a high density of array of electrodes to move just like brain tissue, while also remaining chemically invisible to the entire immune system. And the immune system has evolved over hundreds of millions of years to detect and destroy anything that should not be inside of a normal mammal brain. This is a very audacious goal. Getting a functional brain-machine interface in normal medical context today involves having a large, rigid set of electrodes inserted into your brain. Think of these electrodes as needles that are anchored. Think of these electrodes as a set of needles anchored to your skull. As the brain moves about inside the skull, the section of tissue that's directly adjacent to the needles is continually irritated. The continued tissue damage that occurs at the interface between the probe and the brain will drive an ongoing immune response, as the innate immune system has evolved to recognize so-called damage-associated molecular patterns, which are created by tissue and cell damage. The electrodes will fail to work optimally if the body mounts an effective foreign body reaction, in which the electrode is scarred over in an effort by the immune system to try to isolate the implant as a foreign body and then push it out of the tissue. One of the papers that Musk and his colleagues cite explains the mechanical problem in detail. I'm going to quote directly from the paper. The main hypothesis is that the mechanical strains that arise around the implant site represent one of the leading factors responsible for the sustained tissue immune response. Pulsatile micromotions issued from respiration and cardiac pace create a stress at the probe tissue interface, consequently aggravating body inflammation. In rat brains, these micromotions are on the order of tens of microns for respiration-induced pressure and about 0.2 microns due to cardiac-induced pressure. Tens of microns might seem small, but that's actually quite a bit of movement relative to the size of cells. And here's an image that is roughly to scale. This is cartoons, but I try to make it roughly to scale of a human, a brain, and a cortical neuron. If you're not used to thinking in micron level, a micron is the same thing as a micrometer. It's 1/1000th of a millimeter, which is 1/1000th of a meter. In the middle panel of the brain, the neuron is there to scale, but it's so small and it's almost impossible to see. The full length of the blue neuron is about as long as the thickness of one of the dark pink lines in that brain picture. And in the cartoon of the single neuron, the cell body, the main circular part, it's actually much bigger relative to the rest of the cell than that would be in reality. So here's a picture of the actual dimensions of a cell. On the left is a fluorescent image of a real neuron. This is a cortical neuron in the supragranular insular cortex expressing a fluorescent protein. And on the right is a picture from Wikipedia showing a cortical neuron labeled with a Golgi stain on the top, and then a drawing model of one on the bottom. And the point here being that you can see how small the cell body is relative to the entire arbor of branching dendrites and axons. Chronic brain inflammation is a risky side effect of a brain-machine interface. You really don't want this. This is a super common driver of a lot of tissue disease states that reduce the function of your brain in normal human diseases. That's why brain-machine interfaces are limited to people who are very desperate, where the advantages of the implant outstrip its downsides. The foreign body reaction also limits the effectiveness of a brain-machine interface because when the immune system builds a barrier of scar tissue around the electrodes, then the cells that you're trying to monitor are no longer exchanging electrical forces with the electrode as well. The solution to the mechanical problem is to make the electrodes have an identical mechanical compliance to the surrounding tissue. Compliance represents the ability of a system to deflect upon axial forces brought on it by the surrounding tissues. Bruce Lee said, "Be water." Neuralink says, "Be brain tissue." Think about a piece of seaweed waving in the surf versus a wrecked pirate ship or something that stands still as the waves break onto it and around it. The seaweed is compliant. The ship is not compliant. Compliance is also a property of the object as a whole, which is not the same as the flexibility of the materials that it's made out of. For example, a centimeter thick sheet of paperboard has less compliance than a half centimeter sheet thick of the same paperboard, despite being made of identically flexible material. Also, if the amount of material used is different, but the shape of two objects is different, those can have very different levels of compliance as well. So in order to build something that produces less inflammation from mechanical agitation, Neuralink is trying to make their probes highly compliant and have them kind of blend in with the neural tissue and move around with it and not be like this rigid spear sticking into it that saws against it whenever some little micro motion appears in the brain. So let's go back to the paper and read directly from the next section. An alternative approach is to use thin, flexible microelectrode polymer probes. The smaller sized and increased flexibility of these probes should offer greater biocompatibility. However, a drawback of this approach is that a thin polymer probe is not stiff enough to directly insert into the brain. Their insertions must be facilitated by stiffeners, injection, or other approaches, all of which are quite slow. To satisfy the functional requirements for a high bandwidth brain-machine interface while taking advantage of the properties of thin film devices, we developed a robotic approach where large numbers of fine and flexible polymer probes are efficiently and independently inserted across multiple brain regions. So in order to solve the mechanical compliance problem for electrodes in a brain-machine interface, Neuralink has built the electrodes from materials that, at the thickness used for a functional electrode array in the brain, match the compliance of brain tissue. This presents a new mechanical problem which is that it's impossible to directly insert this tiny flexible thread into the brain in the same way that a rigid needle could be inserted. So the problem they're trying to solve is like trying to stab a length of human hair into a bowl of jello by holding onto one end of the hair and stabbing the jello with the far end, the tip of the hair. Neuralink and the collaborators at University of California developed a surgical robot to insert flexible probes by using a tiny rigid needle as a chaperone for the probe, like a sewing needle chaperoning a thread. The needle provides the required rigidity to break the tissue and leaves the thread inserted when it withdraws from the brain. Again, quoting from the paper, "Here we report Neuralink's progress toward a flexible, scalable brain-machine interface that increases channel count by an order of magnitude over prior work. Our system has 3 main components: ultrafine polymer probes, a neurosurgical robot, and custom high-density electronics, all of which are described below. We demonstrate the rapid implantation of 96 polymer threads, each thread with 32 electrodes, yielding a total of 3,072 electrodes." Here in the final paragraph of this introduction, the high-density electronics are introduced. And those are the final components of the Neuralink design. So the full system includes the electrode threads, a surgical robot to insert them, and the related electronics that will detect and process that signal from the brain. Okay, that's it for now. If you liked the video, please share it, and if you have any questions, please reach out to me on X. I'm happy to clarify anything that I didn't make clear here, or just hear from you if you enjoyed the video. So until next time, be well.